So Paul, we'll just give it till clicks over to 1 p.m. and then I'll do a quick intro. Oh, looks like we're there. Well, on behalf of the full University of Kentucky Shore Resource Center team, I would like to welcome everyone to today's session. If you're joining us for the first time, today's session is part of an ongoing bi-monthly training opportunity series. So we certainly welcome anyone at an R16 eligible institution to engage with us through this series, as well as a variety of other support options that we have available through our center. As always, we are recording today's session and we'll be posting it to our YouTube channel. Please do give us a week or so to get that video trimmed and uploaded to YouTube, however. As we go through the formal presentation, we certainly anticipate and welcome your questions. We would ask that you drop those in the Q&A box. I'll hold those until the formal presentation concludes, at which point I'll share those out so our presenter can provide responses. If anyone is joining us by phone today, we'll certainly allow you to unmute and ask any final questions at the end. So in addition to myself, we have both Shore Resource Center program directors, Drs. Brett Spear and Paul Murphy, with us today. Our discussion is going to be led by Dr. Murphy, who, in addition to serving as Shore Resource Center co-director and an NIH-funded researcher with an active research lab, he is currently the chair of our campus's Institutional Animal Care and Use Committee. And so he's going to speak to you today about vertebrate animal research. So welcome, Paul.
Thank you, Melissa. I'll just say I've been doing animal research for my whole career. I've been on the IACUC here at the University of Kentucky since about 2009. So I've been coming up on 15 years now, and I've been the IACUC chair for almost 9. So I've been doing this for some time, and the IACUC does quite a few different things, and some of which we'll touch on here, but we're not going to go into tremendous depth. I'm just going to try to hit the main points, then we can have some discussion for those of you that may be unfamiliar with the roles and responsibilities of the IACUC and the institution. So here are our objectives for today. These should have went out to you beforehand. I'm going to say the first two here about understanding institutional responsibilities for animal care and the role of the IACUC are tightly intertwined objectives. So these two things work together all the time. So hand in hand, the institution, and by this it means a lot of different things. So there's shared responsibilities between the IACUC and the Office of Sponsored Projects, for example, that are very important. And I will touch on those a little bit. And then the last part here, the third point, is how to prepare the vertebrate animal section of proposal, the basic ins and outs of that, and the do's and don'ts. And I'll hit on some examples and pitfalls for y'all. So this will be hopefully quite a useful session, especially for those of you that may not be familiar with what's involved. So let's start out with something which you would think wouldn't be all that contentious. So what constitutes an animal? Well, as it turns out, there's quite a few definitions. And although you would think this wouldn't be too difficult, different agencies have slightly different considerations and definitions for what they think is important. So some of these are defined through policy and regulations, and some through the actual ins and outs of the Animal Welfare Act and its associated amendments. So this is the USDA's definition. And this includes all warm-blooded animals, and can be varied a bit based on the secretary. But there are several exceptions to this, and these are actually quite important for the oversight of research. So it excludes birds, rats, and mice that are bred for use in research. It includes horses. It excludes horses not used for research purposes, and other farm animals, basically those that are used for food and fiber, livestock, or ag research that's non-biomedical. There's also some additional regulations for dogs and cats that we'll come back to. But you can see that there are some exceptions crafted into here. The birds, rats, and mice exception was meant to streamline a lot of larger scale pharmaceutical research. But as you can see, what's happened over the years is generally everywhere has adopted more or less the PHS model of what constitutes an animal. And this is a much broader definition. So under the PHS guidelines, and this is what NIH follows, this covers any live vertebrate animal that's used in research, teaching, or testing. And note that under this definition, this is also going to cover things like fish and amphibians rather than just warm-blooded animals. Now, It also covers agricultural animals, and these would be ones that are used in research. So biomedical research, for example, under a lot of circumstances, testing and teaching as well. So this is a much more expansive definition of what constitutes an animal and what falls under the regulations. Now, if institutions have a PHS assurance, meaning that they're eligible to perform vertebrate animal research supported by PHS funding, this is the definition that they're following. So they're following the policies and procedures that are outlined in the Public Health Service Policy Manual and the Guide for the Care and Use of Laboratory Animals. So though here we're talking about federal sources, keep in mind that this pretty much covers everything. So most institutions are following this general guideline. So this is also covering agencies that are also federal, but are non-NIH. So there's a lot of shared memorandums of understanding. So for example, NASA, Department of Defense, NSF, they all follow the same guidelines. And you can follow all of this debate online if you want to, but they're pretty much all the same. So there are only a few minor differences between them. For example, Department of Defense has an extra level of oversight where they do a congruence check on the animal protocol, for example. And we'll come back to that maybe a little bit later if we have time. So in order to obtain a PHS assurance number, institutions are required to certify that they follow PHS policies that are outlined in that guide for the care and use of laboratory animals. So the PHS assurance that an institution files with NIH's Office of Laboratory Animal Welfare, or OLA, outlines the procedures and policies that they follow in order to adhere to the basic guidelines and how they follow the law and general broader government principles. So you note here that what you're doing is in PHS, you are ensuring that the appropriate care and use of all animals involved in research training, and biological testing activities is being followed and in accordance with U.S. government principles. That's quite a mouthful. And remember, you are going to get the slides later, so we don't need to read through a lot of the ins and outs of this. But here's what the U.S. government principles say. So proper use of animals, including the avoidance and minimization of discomfort, distress, and pain, when consistent with sound scientific practices, is imperative. Unless the contrary is established, investigators should consider that procedures that cause pain or distress in humans may cause pain or distress in other animals as well. So that U.S. government principle covers a big, broad range of elements for the care and use of animals. But this is critical, right? That this is indicating investigators should consider that anything that's going to cause pain or discomfort in a human is going to be the same in an animal. And that's one of the guiding principles that establishes all of everything else. So that's kind of the umbrella that covers all of IACUC stuff. Now, note also there's an emphasis on the sound scientific practices and principles. So unless established to the contrary, so unless you have a very strong scientific justification as to why you can't, for example, provide pain relief, you have to do so. So what the IACUC does in a nutshell is ensuring that all of the activities, all animal activities in the institution align with institutional policies, and these in turn are derived in compliance with that PHS assurance, USDA registration, and other things such as AAA-LAC accreditation. Now, AAA-LAC stands for, let's see if I can remember exactly what this stands for. I think I had to write it down this morning. Association for the Assessment and Accreditation of Laboratory Animal Care. So not all institutions are ALAC accredited, and some institutions only have partial ALAC accreditation. So for instance, some institutions may have ALAC accreditation for their main animal facility and not for satellite facilities or agricultural facilities, and some will have accreditation for the whole thing. All ALAC accreditation does is certify that you're following certain standards of veterinary practice and so on. And that's actually considered to be more of a prestige thing. So it's not essential for PHS assurance. And it is quite expensive to maintain, so a lot of institutions don't have it. But it's actually kind of an extra badge of assurance, or kind of a quality assurance badge that you can show if you have it. So the IACUC responsibilities cover a lot of different things. And you can see how hopefully some of these align with how sponsored projects might work. I'll hit a couple of the key points here. So the IACUC, for example, will review the entire animal program semi-annually, so twice a year, and they'll report the findings of that back to the institutional official, or IO. The institutional official differs at different institutions depending on their administrative structure. So at the University of Kentucky, it's the vice president for research. That's a common institutional official, but it's not always that person. It can be different people at different institutions, depending on how large they are, for example. This is the person that is responsible for the overall program at the university. So they are the ones that are, basically, they constitute the IACUC, and they delegate the responsibility of oversight to IACUC, and they report back to them. So it's usually not the sole responsibility of the IO. They have other responsibilities as well, but this is one of them. So the IACUC also makes recommendations to the IO about the program. So they're reporting on things like how policy has changed and how they've updated policy, deficiencies that have been identified and how to fix them, and so on. So this is an important communication between the Institutional Animal Care and Use Committee and the institutional official, letting them know how things are going and if any action needs to be taken. Because the IO, among other things, is going to be allocating resources for fixing deficiencies, for instance, so repairing damage to facilities or upgrading facilities and so on. Now, the IACUC also does things like investigate complaints and compliance issues. So these are things that may be reported to the IACUC either anonymously or non-anonymously, compliance issues and unapproved activities that people have done on animals. And they will not only get reported back to the IO, but they'll also get reported to the Office of Laboratory Animal Welfare, and in many cases, the USDA. Now, USDA reporting is a bit different. We'll talk about the difference between USDA covered animals and non-USDA covered animals in a minute. We also do things like recommend suspension of activities involving animals or PI privileges if need be. And probably the thing that everybody's most familiar with is the IACUC's role in reviewing protocols and amendments and authorizing an actual animal activity. Now, one thing I said I would mention here in terms of pre and post award issues is that the IACUC also has a responsibility for determining that federal funds are being expended appropriately to some extent. And we don't oversee the actual accounting, but we do have to interface with the Office of Sponsored Projects, both pre and post award, to make sure that we can track the funding. And I'll touch on that a couple of times going through this. So what needs IACUC review? Okay, so what needs IACUC review is actually somewhat, it's both very simple and very complicated, like most things in research. It can be quite broad. So this encompasses all research, teaching, and testing activities that involve vertebrate animals. They're conducted at your institution or under sponsorship of your institution. This is where it starts to get more complicated. So this is regardless of the species or source of funding. So this also includes activities that are occurring off-site or being performed under contract or by co-investigators at other institutions. And this is something we might best tackle by questions later. So I can give you some broad examples on the next slide. So this is things like antibody production using an off-site contractor. So a large company that makes antibodies that involves animals, they'll have their own PHS assurance. So you would have to verify that it's up to date and that they have their own IACUC approval, and you would defer to them. If you were, for example, purchasing animals that came from a vendor that didn't have PHS assurance, your institution would have to certify that they're following the appropriate guidelines. That can be quite difficult. So, for example, you were importing animals from another country and that vendor in the other country did not have a PHS assurance. That can be quite difficult to certify. So, for instance, our institution doesn't allow that because we can't validate that they're following the appropriate policies and procedures if they're in another country. You may have collaborators or consultants who are performing procedures that are on a grant at a different institution. That institution will have their own PHS assurance, so that PI or co-investigator at another institution would have to provide evidence of the IACUC approval so that you can verify to NIH that that's been done. And this is something you would typically have to do both pre-award and during RPPR. So when you have to update NIH about progress, you'd have to keep that up to date so they'll know that everything is, nothing is expired, for example. Or, for example, someone comes to visit a PI at your institution and they're going to train them on, say, an animal procedure that involves surgery. They would have to be on a protocol and that would have to be approved and so on. So there's a lot of these things that would have to be approved that you may not know of. One thing that I put over here this is one of my guinea pigs, Minimus over here. In addition to being a cute picture, he has his naughty hat on because he bit me on the shoulder. This was just to remind me to tell you that activities are involving privately owned animals do not typically need IACUC approval, but this can actually be a somewhat complicated issue. And I can think of a lot of examples for this, but the one I thought was most straightforward to explain. If, for example, you had a study that involved use of service animals in alleviating anxiety, test anxiety, for example, in medical students. If you were doing that study and it would require IRB approval because it's a human subject study, But whether or not you would need IACUC approval as well would depend on the status of the animals. So if the service animals were owned by the students, so they already had the animals and you were just comparing a group with animals and one without, if they were the privately owned animals that belonged to those students, you wouldn't need IACUC approval. However, you were acquiring service animals and then assigning them to people, that's an animal experiment, and you would have to have an IACUC approval as well as the IRB approval. So with a lot of things like this, if in doubt, it's always better to ask. This is why you have an IACUC and why they monitor regulations for you. So the IACUC can make a ruling on whether or not something requires an approval or not. Okay, so this is a key thing that I want you to remember here. No cost can actually be posted on a grant without IACUC approval that involve animals. So it's an institutional responsibility to ensure that the research that's described in the application is congruent with any corresponding proposals or protocols approved by the IACUC, in addition to ensure that no costs are charged to a grant that have not been approved. And this can impact your institution in a lot of different ways. I'm only going to say this once here because it's a complicated issue, but the issue of congruence is something that trips up a lot of investigators and a lot of institutions because it's a joint responsibility between the IACUC, the investigator, and the institution to ensure congruence. So the concept of congruence is actually relatively straightforward. So when you have a grant proposal that has vertebrate animals and it gets a just-in-time notification, so you have to certify to NIH that it has approval. What the institution has to verify is that all of the animal activities that are contained within that proposal have been approved by the IACUC. And this goes back to NIH and they say, okay, now it's fine to establish an award or release the funds, right? So the institution has to certify that this is in fact correct. And different institutions have different institutional policies for how they handle that. Some institutions will be allowed to accept an award and then establish an account to expend funds related to animal activities following approval, and some will not be allowed to do that at all. A lot of it depends on state laws as well as institutional policies. So that's something that you need to figure out on your own in the context of how your own institution handles these things. A lot of institutions don't have policies for this, in which case there is a default NIH policy, which is actually somewhat similar to how they handle human subjects research. So they'll let it be pending to some point. But again, this is something you need to investigate on your own. We can talk about this a little bit later when we get back to that towards the end. So what I wanted to remind you is that also your IACUC is there to ensure the institutional compliance with regulations. So this is one of their key purposes for being in existence. So because regulations, even though it takes some time for them to be changed at the federal level, because there's so many of them, they do evolve pretty regularly. So your IACUC is responsible for monitoring how these regulations evolve and how they might impact your own institution's policies and making sure that your institution's policies align with whatever the federal policies are and that the institution is in compliance. So you have to use the IACUC to that extent, right? And the other way of looking at this is, of course, is you don't necessarily have to think about this vast array of different regulations because the IACUC is doing that for you, okay? So, and one more thing I'll say about IACUC oversight here before we talk about the vertebrate animals component and how your institution deals with it and how PIs deal with it, is that the IACUC, the best way to avoid being out of compliance with regulations and to be out of compliance with the Animal Welfare Act and so on, is to just do what's in the protocol itself. And that's all we need to say about that at this point. So just keep protocols up to date and make sure they align with what is, what your lab is doing and so on. That's all you really need to worry about. The issue of congruence, when you have to show that your IACUC protocol matches what's in the proposal, only is necessary at the initial award stage. It's nothing that needs to be changed or updated with your program officer, for example, unless something really, really changes about what you're doing in the grant. And that's really complex, and we won't really talk about that much here, but we can talk about it a little bit later if there are questions. Okay, so amazingly enough, the responsibilities for how you put together the vertebrate animals components in the NIH application itself is not really all that difficult. And I say that from someone who has been doing this for some time, but not only just putting them together for my own grants, but also acting as a study section member at NIH and reviewing these for a long period of time, and also reviewing them from the IACUC side, because we typically are looking at these components on the IACUC to establish congruence with what's in your grant application versus what is being approved in the protocol, making sure that all of your activities have been approved. There's actually only 4 pieces on your NIH application that are essentially comprise the totality of what you need to have in that application up front. So I'm going to take these one at a time. So the first one, the first piece that appears in the grant application, is the research and related other project information form. And this covers several very simple questions: vertebrate animals, pending or the IACUC approval date, and then the assurance number. So for each of these different pieces, I'll walk you through what you have to do in the forms, what the potential pitfalls are, and I'll give you some examples. And I'm going to use some examples from my own grant, which are totally straightforward and easy for you all to use as examples. Remember, you have a lot of flexibility in here. There isn't really a total hard and fast format for how you put these together. They're just essential questions that you need to answer. Okay, so the research and related other project information. There is one section here on this page, question 2, which says, are vertebrate animals used? So if you click yes, then you have to say one of 2 things. Is IACUC pending? Or if not, no, which means you already have the IACUC approval and you have to put the date on there instead. And the last bit there is the animal welfare assurance number. So for our institution, our animal welfare assurance number is A333601. So each institution will have their own PHS assurance number. Now, I'll come back to what to do in a minute if you don't have one. So if you answer no to the question, is IACUC review pending, then you have to provide the approval date. Okay. Now, that approval date has to be within 3 years of the application submission date. Otherwise, it's invalid. So my recommendation for most part is to not mess with the date at all, is to just put pending in there, because you're going to have to certify congruence anyway, and there may be some small differences in your grant application compared to what is in the protocol that you're putting in there. So you don't want to run the risk of saying, here, this is the protocol number and approval date for what we're proposing, and then it has some elements missing that you have to amend anyway. So it's easier to put pending and then make sure all of that stuff is in line later when you do the just-in-time submission. NIH doesn't require that approval at the time of submission, but they do require verification of the IACUC before the award is made. And that's the just-in-time stuff that we'll come back to later. Now, remember, all of this stuff takes a lot of time. So don't wait too long. So once you get a reasonable score, even if you think that your grant or application may not get funded on this round, if the score looks reasonable, you should go ahead and get all of that information moving through the system. The reason being is that because it can take time and that time varies from institution to institution, there's very little an institution can do to change things. So if your PHS assurance says, for example, like ours does, that it requires a minimum of 5 business days. If you get an email from your program officer saying that they would like to fund this grant, even though you thought it wasn't going to make the cut, and they said you need to get us your just-in-time information by the end of the week, you may not be able to do that unless you've already started it. So it's always worthwhile to communicate with the program officer to ask them what's the likelihood of funding and see if they can recommend to you one way or the other, whether you should go ahead and do things like get your IACUC approval moving. That can actually be very important. Okay, so this next part here is the PHS 398 cover page supplement. So the most important piece here is the euthanasia description. So this used to be part of the vertebrate animals section in the grant itself, and they moved it up to this part to make it simpler and easier to follow. So it used to be question 4 in the vertebrate animals section. Now it's part of this cover page supplement. So it's no longer required in that section, even though sometimes reviewers are still wondering what's become of it. It's not required there. So here on the cover page supplement is the section that you have to verify euthanasia. So if the protocol or your application requires that animals be euthanized, you have to check yes here. And if you answer yes, then you just have to answer, is the method consistent with the Veterinary Medical Association guidelines, so AVMA. So there are, this guideline gets updated every several years. So you just have to follow the most current edition. And it just tells you different methods of euthanasia and under what circumstances they're allowed and what the proper procedure is for performing that euthanasia. And if you're following those guidelines, all you have to do is check yes. And this is an investigator certification. So they would just have to indicate that they're following the guidelines outlined in the AVMA guide. Now, if they say no, then they have to explain why they're doing it this way. So what I would comment there is if you say no, and it's not consistent, you have only 1,000 characters to provide a scientific justification. And that gets reviewed directly by the Office of Laboratory Animal Welfare. So most people use AVMA guideline methods of euthanasia, and it's actually somewhat rare. So just be aware that if you put that in there, that it's not an approved method, and it has to be reviewed by OLA, that can lead to a delay. You still have to do it, but it will cause a delay in review of the application. But usually it's on the front end, so it's typically not a problem unless you didn't do it correctly, and then it could be a just-in-time delay. So usually it's fine. So remember, this is where you have to put the justification. Now, if you do not have your own animal welfare number or your own PHS assurance number, you do not want to put animal welfare assurance numbers for a project performance side of a collaborating institution. If you don't have an assurance number, your institutional representative can sign, when they sign the application and you have the grant is going to be based at your institution, they're certifying that they're going to get one, right? So when OLA says you need a PHS assurance number, they say they're going to get one. So keep in mind that that's something that they're certifying that they're going to do. If the animal work is going to be conducted elsewhere, the applicant needs to attain an interinstitutional assurance. So for example, you may have investigators that would have a grant based at their institution, but the animal work would be being performed at, say, a contract research organization such as Jackson Labs or Charles River or similar, and they'll have their own IACUC and their own PHS assurance number. So you would just have to have an institutional agreement for how the expenditures would be tracked between the two institutions and who would maintain the IACUC approval. So there are procedures for these things. So for example, your institution doesn't do any animal research and they weren't planning to, there are ways to do it off-site that could be handled that wouldn't necessarily require getting a PHS assurance. There just has to be a mechanism in place that would where you would to establish where you would be doing the animal work. Okay, so this is the big piece, the PHS 398 vertebrate animals section, or VAS. So the instructions for this are actually relatively straightforward. It's usually expected to only be about 1 to 2 pages long, although I've actually encountered ones that are 3 to 4 pages, which are not too much out of range for like larger R1s. It's not uncommon to have 3 to 4 pages. Once it gets much beyond that, so it starts to hit, say, 6 or 7 pages, people will start to get a little bit suspicious, and sometimes you may get a little bit of eyebrow raising because you need to be aware that you cannot use this section to try to circumvent the page limitations within the application itself. And that's kind of a gray area, but you want to avoid administrative review and back and forth discussions with the Center for Scientific Review to determine whether or not you violated the guidelines for how you put together the vertebrate animal section. So again, keep it short, keep it concise. Also keep in mind that NIH may release information at any time through a Freedom of Information Act request or FOIA. And they generally do not resist these because they're not really allowed to. So if a third party asks to see a copy of your vertebrate animal section from your grant, or they do say a blanket search saying they would like all of the vertebrate animal sections from any funded applications at this institute that involve non-human primates over the last 5 years, NIH will just release that. They'll contact the investigators first and they'll ask, is there anything that needs to be redacted? But it's generally, unless you have proprietary information, it's hard to get redactions approved. So keep that in mind when these are being put together. They can end up in the public domain, and they do with some regularity. Now, the first piece here is this description of the procedures in the vertebrate animal section. This is usually the longest part of this vertebrate animal section component in a grant application. The 2 most important things to remember here are, first, you have to include sufficient detail to allow evaluation of the procedures. So anything that's essential to be in the research strategy to understand the science or the project that you're doing needs to be in that section. So you can't bury stuff in this part of your grant application because reviewers may not see it. And this is also where they might actually, you might incur a score penalty because it's in the inappropriate place. So just enough detail to allow evaluation of the animal procedures. Now, there are several musts, so you have to provide in this section. You have to say what the species of animal being used is, what the strains, ages, and sexes are, and the total number of animals to be used. Now, you're providing a summary here of the total number of animals and what they're being used for and what procedures, but you are not providing the scientific justification for the number of animals used. That has to be in the research strategy section because that's considered to be an essential component of rigor and reproducibility. It cannot be here. You can repeat it here if you want, but the key components of it need to be back in the research strategy section. The last thing is if the proposal is proposing the use of dogs and cats, the source needs to be specified. So this is to avoid inadvertent use of, for example, people's lost pets in research. So generally, most institutions these days do not allow random sourcing, although some still do. So random source means you don't have, it's not purpose-bred for research. Most institutions only use cats and dogs that are purpose-bred, but not all. There are some circumstances where that's not true. But if you're using cats and dogs, you need to specify in the section where they come from. Okay, so here's an example of the VAS description of procedure section from one of my own proposals. This is a recent one from the last couple of years. I'm not going to read you through all of this. I'm just going to walk you through the key parts, and you can read it through on your own. So the most important thing is to put the key information up front. So that means you want to knock out those questions right away. The species used, here you can see we're using mice. The strain used, so I've specified the strains, this APP, Delta, NL, PS1, P264 knock-in. The strain, so the CD129 background. Different controls, my general experience with this model, how we breed them, their ages, 6 to 8 months. We use both sexes equally to comply with the NOT OD 15102. And then I tell them below is the estimate of animal numbers. So I've covered the species, the strain, the age, the sex, everything in this upfront component. So I've answered all of those questions so reviewers can find it right away. So they can already check that off and say that this has been done. Then you move on to what the actual procedures are themselves. And your focus here is not on the ins and outs of the science, but on what is actually being done to the animals, because this is the vertebrate animals section. So I will typically split this up into non-invasive procedures and then invasive ones. So the top one here, you see that we've got just monitoring sleep behavior, which is relatively simple, EEG recording, and then we have some social isolation, no extended food or fluid restriction, and so on. So just very basic stuff. For instance, we might do blood samples from them. Things like that could be included up there. So things are generally considered to be things that only cause momentary pain or distress, so like a needle stick or something like that, so relatively straightforward. The more invasive things are things like surgery. So here, there's a minor surgical procedure where the animals are getting EEG electrodes implanted, and then you give the details of how you do that procedure. Again, you need to provide sufficient detail in order for a reviewer to evaluate that you're doing a good job on the animal care procedure end. But you don't need to go overboard in a lot of excessive detail, right? You don't need to talk about what the experiments are because that's not what goes in here. On the plus side, it means you don't have to put things like lengthy descriptions of how surgery is done in the research strategy section itself. You just need to say we're implanting EEG electrodes to measure their EEG activity and establish what sleep stage they're in, for example, here in the research proposal, and explain how you would analyze the data. And the actual procedure itself is in the vertebrate animal section. So it's a good way to save space. But again, remember, do exercise due diligence and caution that you don't want to be seen as trying to go over page limitations by including too much detail in here. So you just need to include enough so that a reviewer can understand what is being done with the animals and how you're carrying out procedures so they can see that you're following good scientific and surgical and veterinary practices. Now, the next piece in there is the animal numbers. Now, remember what I said earlier, keep power calculations for how many animals per group in the research strategy section. So you don't want to put those in here. So you can refer back to that section. So here I said power estimates for group numbers can be found in the research strategy section. And then what you do is you walk the reader through how you get to a total number of animals. And then you sum up by saying, this is how many animals we anticipate that we need. So here I've broken it down by the different aims. So I have specific aim 1 and 2. This is from an R21, so it's shorter. So it says the different experimental groups, at 24 per group, and how many I anticipate will be needed for each of the experiments. And the experimental details are in the research strategy section. This is just summarizing how we got to the number. Now, sometimes you only need a single sentence in the vertebrate animal section to explain where the number came from, and that's fine. This one just has a little bit more details in the derivation. Again, I reiterate here that we have equal numbers of male and female mice because we're interested in sex-related effects. And then at the end, I talk about breeding, how we get to those numbers, and what sort of attrition we anticipate. And then I get to the bottom line number, which is 418. So again, remember, walk the reader through, make it easy for them. So reviewers are busy, they're tired, they don't want to have to search for information. They want to see it laid out right in front of them, even if it sounds like it's really straightforward and silly. Make sure that it's all there and easy for them to find. Now, there are some common pitfalls here. The main one is here, make sure that anything that is essential for the reader to understand the research project has to be in the research strategy section. Watch those page limits or lack thereof. Make sure it doesn't look like you're overstuffing this section. And if you're identifying animal, or if you have animal work being performed at other sites, explain how the animals get back and forth from one site to the other. And again, remember to specify the source for dogs and cats. Okay, the next section is justification for use. These other two sections are actually quite short and really simple for the most part. What you're focused on here for the justification is not the number of animals or anything like that that's in the previous section or in the research strategy section. Here, what you're doing is providing a justification for why you're using a particular animal model and not something else. So why can't you use a cell model or a computer simulation or something like that? That's what you're doing here. And remember, if you're using something other than mice or rats, something that would fall under USDA jurisdiction. So for example, dogs or cats, non-human primates, guinea pigs, gerbils, hamsters, anything like that, you have to put extra effort into the section and justifying why you can't use a non-USDA covered species. So here's an example from one of mine, which is relatively straightforward. Why we use mice, why they're a good model for this, and why we use genetically modified mice. Again, relatively short and simple. If you have a USDA covered species, you need to spend more time here, making sure that it is justified and why, for example, you're using a cat rather than a mouse. So common pitfalls here, if you're using that USDA covered species, you also have to explain why you're not using a non-USDA species. So for example, a common one here is, why are you using rabbits for an antibody generation project instead of mice? Because rabbits are USDA covered species and mice are not. If you're using cats or dogs, the bar can actually be pretty high, and this can often lead to a delay in funding if it's not well justified. If reviewers thought that you didn't do a good job, you may have to write a rebuttal in order to, as part of your just-in-time information, so it can lead to a delay in funding. Keep in mind here that opinions are often strongly held in this area, so take them seriously and remember to address this concern well, okay? So the last piece that you have to address is the minimization of pain and distress. What you're doing here is you need to identify the various types of things that may cause pain and distress in your animals, how you're going to alleviate that or not. So the or not is important. So if you're going to alleviate pain and distress, you have to provide what types of methods you're going to use. So what types of pain relief you would administer for the animals to alleviate their pain and distress. Under what circumstances you would give them pain relief. If you're not going to do pain relief, you have to clearly explain and justify why you're withholding pain relief. And this section often receives a lot of scrutiny. One of the things that's extremely important here is, for instance, if you're proposing multiple surgical procedures in the same animal, why that needs to be done, why they need to be separated, and why they couldn't be done at the same time. And you have to justify that scientifically as well. So here's an example, again, from the same proposal that I had earlier. I typically start this section by outlining general care of animals, so how they're taken care of, What are the humane endpoints? This is something very important to emphasize in this section. Under what circumstances would an animal be removed from the study? So what kind of signs would you consider to be morbidity and mortality? So if the animal looks like it's sick, how would you do this in consultation? Usually it's in consultation with the veterinarian or veterinary staff, and I specified that here. Then after that general care section where we talk about just the basic care of the animals, we'll talk about post-surgical care. So if there's a surgical procedure, how we administer pain relief. And I walked them through this here. So we use buprenorphine or carprofen, and how long it's administered for and what sort of palliative care we administer, how we monitor them for recovery. And then we talk through things like possible adverse events and complications and how we deal with those. So again, remember, be concise, be clear, just make it easy for the reviewers to find the information. Now remember, although it is not necessary to have a section on euthanasia, euthanasia is often discussed as part of humane endpoints, so it can still be in there. Sometimes I still find it helpful to break out euthanasia in case reviewers are confused by it, to make sure that they can find the information. You're not going to be penalized for having extra information. There's only a few sentences on euthanasia in there. You need to have a strong scientific justification for withholding pain relief. That can be challenging sometimes. Generally, what reviewers are going to require, they're going to require both a reasoned discussion of why withholding pain relief is necessary and some references. So keep in mind that the bar can be quite high, especially if you're using a non-USDA or a USDA covered species. It can be a little bit lower in the non-USDA species like mice and rats, but it's still expected, and people expect you to do a good job. Remember, most institutions don't really consider there to be a difference between USDA and non-USDA covered species because the PHS policy treats them all the same. Remember also to justify scientifically if you need to do multiple procedures on the same animal. That also needs to be explained. Now, the last piece here is the just-in-time verification. So this is something that appears in ERA Commons after a grant has been reviewed. Typically, it will often be there even if the score was not great. So that the presence of that button itself in ERA Commons is not an indication of whether or not something is likely to get funded. You need to talk to a program officer, the program officer associated with the application. After you have the summary statements, you have something to discuss with them. They can hopefully give you some feedback on the likelihood of it getting funded or not. Now, the tip here is that as soon as you have a decent score, so if it looks like it is something that has a possibility of getting funded, and this is something that an investigator can determine within a few weeks of getting the summary statement, usually, you should start working on that GIT information because, again, it can take time. And this is how you can really mess up or delay a funding proposal from getting paid out as a notice of award. There are lots of ways that this can go wrong. For example, you can fail to ensure that subcontracted activities off-site have their own IACUC approval or operate under a PHS assurance. Because again, these things can be slow. So if you don't give yourself enough time, even something that only takes a couple of weeks can set you back significantly. You want to make sure that you can adequately explain the use of chemical hazards, select agents, and biohazards around animals and address issues of safety. Now, some of these issues are not addressed in a specific section. So there is no specific section in your grant application that addresses biohazards, but it is a scored requirement. It doesn't get its own individual score, but it's factored into your grant overall impact score. So because biosafety approval and safety approvals in general and chemical hazard approval may operate on a different cycle from IACUC approval, keep that in mind that can delay approval for just-in-time submission to NIH and could delay funding of a grant. And that last one that I mentioned was the issue of failure to address congruence. So because this can actually lead to another delay, make sure that you give your IACUC enough time, or your institutional officials or your office of sponsor projects or whoever the person is at your institution that's validating congruence, that they have enough time to compare the IACUC protocol that's being approved with the contents of the grant itself. Because as you might imagine, grant applications can be complicated, and the people that are going to be evaluating congruence are just looking to see which things match and which things don't match. And it may require substantial back and forth with those individuals because they're probably, actually, I would say almost certainly will not be aware of the ins and outs of a specific research program, and they'll need to be, they'll have questions, and you'll need to have time to answer those questions and resolve these things. So a couple of last words here. So your vertebrate animal section, the vertebrate animal section is considered in the score when reviewers rate an application, but it doesn't have its own score. So that means that having a good, well-done vertebrate animal section in a grant application is essential, but it's not going to help your score, but it will hurt it if it's not well put together. So keep that in mind. You have to answer all of those questions adequately, and then the reviewers will just say that the section is adequate. So I know not very satisfying, but I mean, if it's inadequate, then it lowers your score and you have to address those. Now, remember also that regardless of when experiments are planned in a project timeline, so even if the grant is 3 or 4 or 5 years, NIH requires all the activities be approved as an award condition. So even if you don't anticipate that a particular experiment might be done until year 4, and the IACUC approval may be for only 3 years, you still need to get approval up front. Now, remember again, when I said that institutional policies differ, so you need to know what your own institutional policies are for accepting an award and how accounts can be established and when money can be expended. Because in some circumstances, you may not be allowed to accept an award, or NIH may not be allowed to issue an award without the appropriate approvals, right? So you need to understand the timelines for these things and what is required of you to make sure that you get these things done, because you really don't have very many options to make things go any faster. So you need to plan accordingly for giving yourself enough time to get these things done and uploaded in time. And I have plenty of time for questions, so I stop here with a cool picture of my cat.
All right, thanks, Paul. We have a couple of questions that are in the Q&A already, so I'll start with those, but if others come up, certainly feel free to continue populating them there. The first question is in reference to the IACUC protocol number justification section. It says, I'm conducting a feasibility study using rat models and do not have data from previous studies. So for feasibility studies, how do we justify the number of animals? Do we always have to follow the power analysis method for determining the size of the sample? If so, does this apply to the feasibility studies as well?
No, you do not always have to use power. So if you don't have preliminary data to derive your own power analyses, what you do is you use published references that studies that are similar to estimate the animal numbers. So you would find a similar rat study that had similar procedures and say, based on these published studies in peer-reviewed journals, I estimate that I would need this number of animals. And that's an accepted method to estimate animal numbers if you don't have an actual power analysis present. But you do have to include something that has to be in the research strategy section.
Okay, great. The next question says, for an IACUC protocol, for adult rats 23 months old, when using carbon dioxide for euthanasia, do we have to add the methods to ensure the animal will not revive?
So in your IACUC protocol, you do. So CO2 asphyxiation for rodents is a conditional. So you have to have a secondary method for euthanasia. So this would be something like, for mice, it would be cervical dislocation or decapitation. It could be something like thoracotomy, or usually cervical dislocation isn't a good method for rats, but decapitation would be, because people have difficulty cervically dislocating rats. But you do have to have a secondary method. That's not something that you need to put in your grant application, though. That method of CO2 asphyxiation with a secondary method is in the AVMA guidelines as an accepted method of euthanasia. So you would just say yes in the grant application itself, and that would be fine. In your IACUC protocol, though, you would have to specify what the secondary method is.
All right. Well, those are the questions we have at the moment. If anybody would like to request permission to unmute and ask a question, we can take those. Otherwise, we'll just stay online for another minute or two in case anything else pops up. For those of you who are interested in receiving a copy of the slides, I will pull the email lists from the registration data and send a PDF copy of today's presentation to that email address. If you don't receive it in a couple of days, you are certainly welcome to reach out. All right, well,
I think someone raised their hand.
good catch. I just saw it. All right, Jennifer, you should be able to unmute now.
Okay, thank you. Yes, so we had a very experienced researcher who had his IACUC protocols going through approvals, no problem, but he got hung up with IBC. And we didn't even know that he was needing IBC approvals. So I'm a research administrator. How could we have helped make sure that that was also in the works? Any recommendation to the administration side? Because now we're delayed significantly with that piece.
So we have that happen fairly often, actually. So we have on the IACUC approval side, we actually have a requirement for the Institutional Biosafety Committee and Health and Safety as well to sign off on protocols before they're approved. Because in the past, we actually had issues where people would get their animal protocol approved without their IBC approval, and we wouldn't know that that was true until later. So now what we do is we have an administrative hold that can be put on, so the IBC or occupational health and safety, environmental health and safety rather, can hold a protocol until they approve it. So they don't get the final IACUC approval until they have all of those things done, and that's how we prevent that from happening. And that seems to be the most efficient way. The reason being is that most institutions don't have environmental health and safety, and IBC typically don't have, at least in most institutions, don't really have the teeth to enforce regulatory compliance, whereas IRB and IACUC do. So they kind of work through those to make sure that that compliance is maintained. That's the best way to do it. Now, in terms of what to do with a PI that happens to, when we've had these sort of miscommunications happen in the past, because NIH just requires that the IACUC approval be secured, right, specifically as part of the guidelines, what you can do is NIH can still make the award, and what will happen is sponsored projects would hold the award and not issue an account until all the compliances are in order. But that, a lot of that depends on what the institutional policies are, because our institution no longer allows that, but many do.
Okay, that's helpful. Thank you. Yeah, it was the latter situation, and we're working with OSP. So I will continue to make suggestions.
That's kind of the default NIH policy, that they'll allow you to issue, they'll allow the award to be issued. So you get the award issued in time, just they can't expend any funds until the regulatory approval is acquired.
So at least we can move the notice of award forward, get accounts set up.
Because if the IACUC approval is already done and it's congruent with what's in the application, then they can issue the award.
Yeah.
Because the regulations are different for IBC and health and safety approval.
Yeah. And the IACUC was contingent on the IBC being resolved, but they did want it to move forward. So
yeah,
everyone's trying to make it.
Yeah, there's different institutional policies, right?
Yeah.
So I mean, some of them, our institution doesn't allow that anymore. And the reason we don't is because we had some issues where it kind of fell through the cracks and people expended money without getting the approval, and that created a problem. Right. So I just don't allow it. But I mean, lots of institutions do, and that is allowable within NIH's guidelines.
Okay, interesting. Well, thank you for the presentation. It's excellent.
Thanks.
All right. I don't see any additional questions or hands. So if there isn't anything else from the participant group, we'll thank Dr. Murphy for an excellent presentation. And again, I will be in contact via the emails that you registered with to share a PDF copy of the presentation shortly.
Thank you all for attending.
Thanks all.